sure enough, we put the strips in and we were able to keep the bees alive. And then those strips started to fail, and I tried camouflage for 1 year. I go, oh my God, if this is what beekeeping needs, and I got to put, organophosphate, this long residue to my beehive. I'm not interested in it. And so at that point, I stepped off the synthetic chemical side conveyor belt because I just thought it would just keep going. I thought that amateur apps would fail just as fast. It hasn't done that, but it is now failing now. So it was a hard learning curve, learning how to control varroa. Way back then, I had to follow most of the research from Europe because you guys are ahead of us on this. So Europe is ahead of us, then there's us, then Canada is about 2 years behind us, and New Zealand was about 10 years behind us, and Australia now is 30 years behind us. So when I go to like New Zealand, it's like I go, it's like deja vu. We don't even use those questions. You guys must have been on the internet. Because check the date of those posts. You don't have to repeat all of our mistakes again. So I thank the European beekeepers that most of the research I followed on varroa was from European, because you guys had it before us. And so then it's the learning curve for me to adapt those for the United States. And you guys keep these very different than we do. But that's more a function of the human influence than the bee influence. The bees are still influenced. They're still pretty much the same. So just because your beekeeping methods are different, it doesn't mean that your biology of your bees are different, or that the biology of varroa is different. It means that the beekeepers are different. So I'm not making any recommendations. I'm not suggesting that you change your methods, because what I said, I'm just showing you what works in my operation in California, what works in the United States, where I'm familiar with. I don't propose that I know anything about keeping with AC hives or beekeeping in Slovenia. So it's up to you to interpret what you see from me to apply to your own operations. One thing I tell people is, oh, we got to help the beekeepers, we got to do this. I think you don't got to do anything. Most businesses, 90% of businesses fail. Okay? It's not my job to keep everybody who wants to try to make money with bees to do it. You can't cure stupidity or anything like that. There's going to be a lot of people who start off, and it's going to be it. And in the U.S., I tell beekeepers, hey, you're all my competition. Every tip and trick I give you makes it harder for me to make money because you're going to be more successful. Okay, so that's the bottom line. So I'm not on a mission to make every beekeeper be successful and hang away. Okay, but those who are interested in good, reliable information, I am happy to share that with you. Okay, so understanding varroa, sustainable management. So like these rotating these organic treatments and eventually mite-resistant bees, that will be sustainable. This will be the most sustainable. This is stopgap until then. And then as the climate changes and we've been in this 20-year drought, worst drought in a couple of thousand years in California. We're 20 years into this, and it's just learning how to use colony nutrition and supplementing. How many of you in this audience feed artificial diet, pollen sub, or any kind of protein to the bees? Okay, just a few. So as your climate changes and there's less and less forage, and you get more beekeepers, you're already over carrying capacity, you may need to start taking pollen sub. And I was starting out with bees. My first quite a few years as a hobbyist, I said, oh, I will never feed sugar syrup to a beehive. I'm not going to feed pollen sub. That's unnatural. It's artificial. And then after a number of years, a commercial beekeeper became a good friend of mine and said, Randy, I don't know whether sugar syrup is good or bad or this or that, but I can tell you one thing. You give a colony a gallon of sugar syrup at the right time, it's like waving a magic wand over that colony. It can really make them much healthier or survive. And I realized, yeah, that is true. So I kind of got off that, oh, I'm so natural bandwagon and said, really what I care about is managing my livestock and keeping them healthy and productive. So for 25 years, I picked up all my beehives from when it dried up in first week of July and on trucking over the mountains up to an irrigated lucerne in the state of Nevada, where they did much better. And then there was a plant that bloomed in the fall that produced good pollen for wintering. And then I would haul them back. As Varroa started to take its toll, it got harder to manage those bees when they're a few hours drive away. So we started experimenting with feeding pollen stuff and learned how to keep bees healthy at home. So now, yes, we are feeding our bees a bit of artificial feeding in the late summer, but we're also home for dinner every night. And I like that. I don't have the carbon footprint of all that pollen of hives. So let's talk about using pollen stuff. And I snipped together out of some much longer presentations, this one right here. So you can hand mix it. We also have mechanical mixers that we use for it, or you can buy it pre-packaged from the various manufacturers. You guys don't have small hive diddler yet. I didn't realize that coming here, so I put this in here. If you are feeding pollen stuff, the bees will only consume it if it's placed within the cluster. Anything that hangs over the outside of the cluster, they're not going to touch it. And then that becomes a magnet for small hive beetle and breeding ground. So if you're going to feed pollen stuff, only put it in the middle of the cluster. Don't put it on the top of the hive. Put it between your two brood chambers, as close to the nurse bees as possible. The nurse bees do not respond. Okay, first thing to understand if you're a beekeeper, everybody knows that perfect frame. You pick it up, there's a band of honey to the sides and across the top, then there's that nice arc of bee bread, and then there's a brood in the middle. When a forager bee comes back to the hive with a load of pollen, where does it preferentially on that comb place the pollen? Where? In that band around the brood, or where? Where's the forager preferentially place the pollen? On that frame. Here's the frame. Does it place it up in the honey? Does it place it in that band of bee bread? Or does it place it in the brood? So it places it preferentially right in the middle of the brood nest. And that pollen is then consumed very rapidly, often within an hour, by the nurse bees. That's where the main dynamics of incoming pollen are, right in the middle of the brood. Anything you see in that band, that's surplus. That's what they don't need, the leftovers. And that's like that cupboard in your house with that canned food that you saved for a rainy day. That's not stimulatory at all. You don't go in there because you want to eat that stuff. That's that sauerkraut that grandma gave you 10 years ago that you're storing for a bad day. That's how the nurse bees look at that band of bee bread. It's fermented pollen that they'll eat if they have to, but that does not stimulate brood rearing in any way. If you want to stimulate brood rearing, you got to put that pollen stuff, or your natural pollen, as close to the center of the brood nest as you can. So there are advantages of using unwrapped chunks rather than the papered chunks. What we do is we get both, or we make both, and we chop it up with a spade, we put a big chunk in, and we feed over 1 kilogram at a time. Okay? A hungry colony will eat a lot. Many beekeepers underfeed pollen stuff. That's just an appetizer, a small amount. You want to give a good sized chunk. We press it down, and we want to squeeze between the frames, and it's right in proximity to the nurse bees, and it's very stimulatory to them fed that way. And the other question is, what do the bees do with it? And I didn't put the pictures in here, but we've done studies with, a lot of studies with fluorescent tracer again. And what we found was, they don't store any of it as bee bread. They consume it or they throw it away, but none of it gets stored if it's fed in this manner. Now, if you put dry granules of bee-collected pollen in a hive, some of the bees will pick it up, actually put it on their legs. And then the funny thing is, if you're feeding it on top of the hive, so I put a rim on top of the hive, I put a piece of glass over it, and I put petri dishes of pollen stuff and pollen to see what the bees would do with it. And when you initially put that powdery pollen in there, you'll see a bunch of bees come in there and get it on their bodies, comb it up, put it on their legs, and then you think they just walk down a couple inches deep to the nurse bees. If you crack that glass, they will fly out, turn around, fly down to the entrance, come in the entrance, and then come back up. Because that's their behavior. They come in that way. So they will, if glass isn't cracked, they will go down, and then they will pack it into the cells, and then the nurse bees will eat it. But it's the nurse bees eating it in patty form, where it's moistened. They don't store any of that. So you can also dry feed pollen stuff, and the foragers will just mob this. They will cover their bodies with it, comb it with their legs, and bring it back. They will store that in the colony. They will make beet bread out of it. The problem is it's really erratic, that out of one hive out of 10 will have every forager going to it, and the next hive won't have any foragers going to it. So it's very easy to do because the bees are colored with the pollen stub. So you walk around your bee yard and look at the entrances, and you can see some hives with every bee coming back with pollen stub, and other hives with none getting. So what we have found, that's an easy way to feed, but it's very erratic. So we gave that up, and we actually put a measured patty in every hive. Because we want to treat, we don't want to baby any particular hives. We want uniform hives, so that everybody's the same. Any that are not the same, we either combine them or get rid of them or something. We want to have all of our hives about the same for our management. And then generally, you're going to need to feed some either nectar or sugar syrup because the nurse bees need a lot of water in order to create the jelly. So just feeding them protein, they also need a lot of water and some nectar or sugar syrup that apply for the sugars that they have to also feed to the larvae and mix with the jelly. So generally, you're going to feed 1-to-1, one part sugar to 1 part water for stimulation to get the nurses to rear brood. You're going to feed 2-to-1, 2 part sugar, 1 part water for fall feeding or anytime you want them just to put on stores in the hive. Now, all the other livestock industries followed up on the work of those scientists who worked at the same time as the growth back in the 1950s. And every other animal industry got us way ahead of us as far as developing artificial diets. Almost any, you can buy a specific artificial diet for raising ostriches, or raising alligators, or raising weasels, whatever you want to raise. And not only that, if you're raising chickens or cattle, you will have one artificial diet for their first week of life, and then another one for the next 2 weeks of life, and another one for the next month. And they change the ratios of the amino acids throughout those, and maybe the vitamins and the minerals, because these nutritional needs, when you're growing bone, you need more calcium. Once you've grown that bone, now you're going to need more protein for muscle mass, Then you grow hair, so you need a different amino acids to grow the hair. So we haven't developed that far. We are so far behind on honeybees. So the question always is, there's a bunch of people selling pollen stubs, and everyone they claim to be the best, okay? So the problem is, we don't have, you know what the magazine Consumer Reports is? Okay, in the US, there's a magazine, it's non-profit, and if you want to find out what the best toaster is, or the best, best frozen food is, they buy at their own expense, through their subscribers, all these products off the market, and they run controlled tests and they compare them, and then they report the results. We don't have a Consumer Reports for honeybees. We don't have independent testing. If you're raising roses, they have the All-America selection. So if you come up with a new cultivar rose every year, they have a whole bunch of volunteers and they plant a bunch of different cultivars in different climates, and then they test them all, report back and say which ones perform best. That's the All-America Rose Selection. Okay? They have it for other plants too. We don't have that kind of independent testing for honeybees. It's really unfortunate. So, yeah, we have me. So I do a lot of independent testing of products that come on the market, including the probiotics. I've got a bunch of data on that too. And I agree with Ivana that most of them on the market are, I'm going to be publishing a whole bunch of data with next-gen sequencing where we really followed it. We treated them with antibiotics, look for recovery, look for build-up, weight gain, look for quality strength, look for pathogens, but decrease the pathogens. They did nothing because they didn't use the species of bacteria that actually will colonize the bee gut. So you can't use an animal probiotic that doesn't do any good. Now, not to say that there couldn't be any probiotics. There could very well be if they did the next generation and actually cultured the specific strains of bacteria that live in the bee gut, so it could work. Anyway, so every manufacturer says, well, ours is the best, but they don't show much data. So I have, over the years, run a number of trials to compare them. 2013, I ran one testing most all the pollen subs that were on the market with a negative control of just sugar, equine sugar alone, and then patties made of natural pollen right here. And then all the tested subs fell between the natural pollen and the negative control. And the negative controls typically, you can see, they go from 5 frames of these down to 2, they're almost nothing, they won't make it to the winter. It's like, I intentionally put them in the worst yards where there's no natural pollen coming in. So that was 2013. We found that some of the top tier subs actually performed very well, but one of the problems is when I analyzed this, I found out, well, that's because they had a higher protein content than these right here. So in my subsequent trials, I then matched them for protein content to see. And so I've run other trials I'm not going to show you. I'm going to jump ahead to my big trial in 2020. And the reason is that this is what California Central Valley looks like right now. The almonds are in bloom right now. And what you have is let me see if I have it. No, I didn't do it. If you notice, each row of trees is a different color of flower. So they plant different cultivars, they're all self-infertile. They can't pollinate, they're all clones in a row. So they cannot, one tree in that row cannot pollinate another tree. You have to have bees carry the pollen from one row to the other. And the growers pay us a lot. I started out almond pollination getting paid $10 a colony to rent. We're making $225 a colony. Now, in the years that I've been doing this, the price has gone from $10 to $225. That changes everything. And the growers grade our colonies for strength. We get paid by how strong they are. So producing strong colonies by February is really important to us. And first thing, a strong colony requires a good cluster going where this cluster would probably be too small to make top grade. If this were a fall cluster, that would be too small. They have to be really big going in. Because in January, when they start building up, the queen's egg laying is limited by how many combs are covered with bees to give them more cells to lay the eggs in. So you have to have a large cluster to start with. So that means we need to get pollen sub in the fall to get a large, well-nourished cluster of bees going into the winter. And this is what our environment looks like all from July on. There's nothing for the bees to eat, and there's no fall bloom. So we feed pollen sub, we control varroa first in August, and then once we have the mites down to 0, so you don't have viruses, then you start rearing more the bees of your winter cluster. There's no reason to initiate brood rearing if you have a high mite level because you're just making more mite food. So first eliminate the mites and then start feeding pollen sub in the fall. Now, if you're a hobby beekeeper and your main concern is swarming, you don't want to feed pollen sub in the fall because you don't want a great big cluster because otherwise February you're going to have pollen swarming. So you have to look at what your goal is. If, on the other hand, you want to sell a whole bunch of nucleus colonies in the spring, then you want a big cluster going in there. You want to grow them up so you can have bees to split or sell come springtime. So just because we do this, and we do both of those. We pollinate almonds and then we sell 1,000 nucleus colonies, okay? So if you're the hobbyist and your main concern is just having some bees and not having them swarm, then you may not want to keep pollen subs. So when we take our bees down to the almonds, they want the growers want to make sure you actually are going to come through on your contract. They say, can you get the bees down here? We go, we're on a pollen and nectar flow up here in the foothills right now. They're doing great. You want us to bring them down now to the desert? It's a desert for bees. There's nothing for the bees to eat. So we'll bring them down and then we'll put 1.5 kilos of pollen stuff on them because they're already rearing brood based on natural pollen flow. We want them to continue that brood rearing until the almond trees start blooming. So those are my two sons doing that. And we are facing more droughts like here. So if you look at drought intensity, the more intense the drought, the more red and purple it is. This is averages from 1950 to 59. You don't see it very much there. Averages from 2000, 2009, we're starting to see droughts. Okay, so where are you guys at? Right around here. Look at expected for 2060 to 2069. Some of you guys are still going to be around here. It's not looking good for you. No. You know this. Okay. So that means you may want to learn about supplemental feeding. So in 2020, I wanted to answer this question. Anytime you do a research project, specify the question that you want to answer. And this one, do some pollen subs, you can't say are they good, what metric? So our metric is to promote colony growth to get how many frames of bees that I get paid. We get paid $25 per comb of bees going to almonds. So a very simple metric, dollars into my pocket. How many $25 payments do we get by feeding one pollen sub rather than another? Okay, so this is from the 2013. By 2020, we had 2 new ones on the market, and some commercial beekeepers had developed a homebrew formulation that they liked and said, Randy, we're curious how our homebrew formulation, which we can make very cheaply and looks really good to us, how will that compare to the products in the market? And they gave me some money to help fund this program. I had some of the one of the pollen stuff developers say, hey, Randy, they are $50,000. Can you test my product? I said, well, I'll test your product all right, but it's not going to be on your money. Okay, I want the beekeepers to pay for it so that we see. So, so this is the boat saw patty made by Man Lake, which we have used for quite a few years and works pretty well. Although this year, when we tested it, they changed the yeast, and the bees hated it. So it wasn't a fair test. After I ran this, they changed back to the other yeast, so it actually works better. So this actually worked worse in this year than we expected. Another company makes a very common patty, and we got the one with the 15% natural pollen in it. Data makes, this is their new one, artificial pollen 23 is what they call it. And developed by a nutritionist who I've corresponded with quite a bit. He's helped me out too. Then a sugar patty, a fondant we made to match roughly the amount of sugar in these, but with no protein in it at all. Mega B developed by Gordy Wardell from USDA many years ago. Ultra B, Man Lake's flagship, which is very similar to this, but in patty form and a little more expensive. And then the homebrew with 20% natural pollen from the commercial beekeepers. And then this new one with spirulina algae in it. But these guys also tried to control varroa and added a bunch of thyme oil, which the bees hated. So it performed very, very poorly in this trial. After this trial, they switched and they took the thyme oil out of it. So if you have small hybrid, the thyme oil thinks the small hybrid doesn't like it. But if you want your bees to eat it, you take the thyme oil out. Here's my son Eric. We ran this trial with 144 hives, 18 hives per each of the patty types. We replicated in 3 separate yards to see when you got different results per yard. It's always good to replicate any trial of honeybees, either in different years or in different yards, to see if you get the same result. And in one trial, one time in 1 yard, you you can't count that that's going to be consistent. Now, this was confusing. We had to make a bunch of feedings with all these, there was seven different types, plus the sugar, so 8 altogether for the sugar control, and not make any mistakes. We couldn't feed the wrong one. So every day we went to feed, we would count out in color-coded tubs 18 patties, and we put a color code on each hive. We didn't care, we didn't pay attention which was which. We couldn't blind ourselves completely, but we didn't pay attention to it. And then we lay out the patties on top of every hive, and then a quality control person would walk around, double check to make sure they were all laid out properly, and then we would crack the hives and put them in. We chose yards, this is my 2 sons, that we knew the bees were going to crash it, that there was just nothing for them to eat from our experience, okay? And we fed continually pollen seven syrup from late summer into winter and cut up. So we started feeding in August, took our first grading of them, fed pollen syrup and seven syrup. So pollen syrup feedings are the yellow boxes. Feedings of syrup are the round circles. And we got this unexpected yellow star thistle, Centaurea, and you guys may have this around here. It gets 10 days to produce pollen and nectar. We weren't expecting that. So we did not feed pollen sub, and that was in only 2 of the yards. So we skipped those 2 yards then. Then no feeding, we graded midpoint November, stopped feeding. So this is all birth period. And then starting middle of December, we started feeding again. And then we had a natural flow from alder trees. So once that started coming in, we had 21 days before it affected emerging bees. It hadn't affected alder trees. So 21 days after the alder flow started, we did our final grading before going to almonds. So this is typical of what the combs look like in mid-October, just before going into the winter cluster, only from artificial feeding. But notice there is no stored bee bread, as there was no natural pollen coming in. The only nutrition they were getting was strictly from the artificial diets being fed to them. And then we did our November midpoint grading with my newest technician, who was Eric's son, my grandson, who was out of school due to COVID, and well, he was sharp for a quality control recorder, and he made sure we got everyone right with our right color and get the numbers down there. So we picked the boxes up, we pick up both, split them apart, grade from the bottom of the bottom box and then from the top box, and very accurately grade for strength. Okay. So, this is at midpoint grading. This is the November cluster size of frames of bees. Now, it was a cold day, so the clusters were shrunk, and they were pretty small. Now, I'll tell you right now, we had this extremely hot, dry summer. Most of the colonies shrunk in size. They were smaller. The August grading in hot weather, the clusters were larger. But when we graded in November, it was cold, so they were small. So, but it doesn't make any difference because we were grading relative to each other. Okay, so we don't go by the absolute value, we go by the relative comparison in strength. So this is our colony strengths of the different color-coded patties. Now take a look at the red one, which was the global, and the home group, because they were exceptional there. The other thing to look at the error bars. Look how consistent. There were 18 colonies in each group. 18 hives, and look at how small the error bars are here. When you have small error bars on every group, that means that data is probably pretty good because you're so consistent on the small error bars there. So we went into winter like this. Now what was interesting, I often collaborated with university or UC, Agriculture Service, USDA researchers who have the equipment. So I sought one out and said, hey, can you pay for the pollen 7 chemical analysis? Can you also then look at bee weight, dry bee weight, and can you also then do next-gen sequencing on the gut microbiome? Okay. Okay, so I did all the field work. he did the lab analyses. So here is, on the left column, that is the total frames of bees in the group for all 18 hives. The right that's the right column. The left column is your head and thorax mass. Okay, so that includes the hypopharyngeal glands and the flight muscles. Okay, look how closely they correlate. So what this tells you is that that difference was due to the colonies getting greater nutrition, and you got heavier bees, more body mass with the better nutrition, and that also then meant you had more frames of bees. So total number of bees in a hive correlated to how well fed they were and how large they were. So now you want to wonder, well, what variables are there in these pollen cells? So let's look at the protein content. They vary from 22% down to less than 15, and there's no correlation between protein content and performance. Keep looking back to the two starters. Fat content. Two of the lowest fat contents were the best performing. Okay? And by the way, the critical sterol, which is in the fats, 24-methylene cholesterol, when I had this one right here was 0. Okay? So even though it lacked that one, the bees were able to synthesize their own and do okay. Sugar content. We analyzed the sucrose, glucose, fructose, and total sugars would be the solid color right here. Nothing special about sugar content, okay? Relative prevalence of the core gut bacteria. So the height of this is total number of bacteria, and the different colors are your core groups. So you can look at the relative percentages of them, and the species composition was the same, whether they were fed pollen or not. But some of them had more. So let's look here. That one was 20% pollen, that was 15% pollen. No correlation to the amount of natural pollen. You guys get an idea of how fun bee research is? Everything you think is going to be make sense to you? Yeah, throw that away. And then, since I had some of my resistant breeders, potential breeders that we did not treat, we treated all the rest of oxalic acid, I said, wow, let's take a look to see if oxalic acid made a difference in the gut microbiome. That's good data, didn't make any difference. So, the answer to which variables correlate with performance, we had no idea. We threw everything out the window. It's got to be something else. So, in December, we resumed feeding again, and again, no natural pollen. This is all based upon the initial feedings of these overwintered colonies, artificial diet again. We did our final grading on a very, very cold morning on February 1st, just above freezing, with very tight clusters. Final results. So this line, this is your endpoint strength frames of bees for each colony, and this is your midpoint strength, okay? So this is showing growth from the winter cluster up to final grading. This would mean no change in strength right here. So if they're above this line, that means they grew. If they're below this line, that means they shrunk. Look at the colors. Do you see a pattern? No. Which color is more prevalent above the line? The home one, yeah. And the blue. The global and the home one. Which ones are more prevalent below the line? Yeah. The healthy bee and the native control, lots of white, lots of green. So we're already seeing a big difference in growth right there. Now, was it consistent from yard to yard? Yeah, it was. All the yards were pretty much the same. The colony started out averaging 8.5 frames of bees in August. They averaged out down weaker in November during that cold cluster, and then some grew, and the native controls continued to shrink in size. They not only did not grow, they continued to shrink. Did it have to do with pollen? Well, these two did have pollen, but this one had 0 pollen at all, performed almost as well. And the thing was, this was our favorite book, soft, when they switched the yeast in. If they had switched the yeast, my guess is that this one would have been right up to the 2 of them with no pollen. So my take home is, I don't see that any natural pollen is necessary in any way in an artificial diet. We'll get to it in a minute why. Now, how about colony loss? So this is percent of colony losses. Didn't lose any colonies in global. We lost 30% with the sugar control. Okay? Now, just for a histogram of comparison, if you're going to compare, I'm not going to give you a bunch of numbers for statistics. Your eyes are as good as any statistical analysis. You look at the histogram, the distribution of 2 different groups. So this means out of those 18 hives, these columns will add up to 18 for each group. It shows, on average, the sugar control colonies average 2.7, median of 2.9 frames a beat. Very, very weak. The homebrew, both the mean and median were 8.3 frames. If you look at these 2 groups of hives, you can see, you know these statistics. There was a big difference based upon pollen sub type. All statistics does is just come up with a probability value that researchers spend days trying to tease out a p-value of less than 0.05 for probability. But this you can see very strong, very low probability. Okay, final analysis. I said, well, easiest thing to do is out of those 18 hives, how many total frames of bees were there that would pay me $25? And the home brew. We made good money on the home brew. 3.5 times as many frames of bees in the home brew group as there were in the sugar controls. So a very clear difference of what you're looking at. Now, step the question. Why did some subs perform better than others? Now we start the deep dive. You all go, oh no, not again. So we get to the concept called Liebig's barrel. And Liebig's barrel says any living organism, whether it's a plant or an animal, requires a balance of nutrients for optimal growth. And he says, imagine that all the nutrients are in balance, and each stage of the barrel represents one nutrient. One may be carbohydrates, one may be iron, one may be zinc, one may be the amino acid tryptophan, one may be vitamin C. If any nutrient is out of balance, deficient, that's called the limiting nutrient, and that stage of the barrel is lower, and that determines maximum growth. Doesn't make any difference how high the other stages are. Your growth is limited by the first limiting nutrient. Okay? And that's where we're going to go from now on. Figure out what the limiting nutrient was in these pollen subs that prevented them all from growing optimally. For example, I noticed you guys have dandelions blooming already. We don't have dandelions blooming yet. You guys are having a very early spring. Now, bees love dandelions. If you put bees out on just solid yellow dandelion flowers, what will happen is all those colonies will bloom and die. Because dandelions are missing short on 2 essential amino acids and not enough to sustain growth of the colony. So on dandelion pollen alone, colonies will wither away and they'll die. Same with pollen stuff. If you have a limiting essential amino acid, you're going to wither away and die. The state of our knowledge of pollen subs for honeybees was published in the Netherlands by Antonius de Groot in 1953. I was 2 years old. Okay? And everything we know is based upon this graph right here. This one single graph. And the numbers that we have for the growth recommendation are based upon the inflection point of these curves. See how the inflection point? What that means, he fed these adult bees in cages, an artificial diet made solely of essential amino acids, and at either 1% concentration, at 2%, at 3%, at 4%. So if it was deficient in it, that means if you add it, it will increase the amount of weight gain here. Once you reach the ideal amount, there's no added benefit to adding any more. So you compare, this one would have a value of 4%. This one would have a value of 2%. This one would have a value of 4.5%. And tryptophan just happened to come out at a value of 1%. And for that and other reasons, the growth said, oh, I'll just normalize the tryptophan because it's got a value of 1. Everything else would be some multiple of tryptophan. So he came up with this whole 8-page paper was all about ratios until figure 15. And he made the terrible mistake of saying percentages. And he should never have done that. Because that's the only one anybody pays attention to. And so they all publish, the black line would be the minimum percent in a diet that the growth recommended. But the growth, they don't read the rest what he said. He said, this is not for optimal growth. This is for absolute minimum growth, much less than you would get on natural pollen. And when we analyze these diets for all these 10 essential amino acids, they all were above the growth's recommended value for minimal growth. So all the manufacturers said, yes, these are complete diets. They're really good. And I looked at this, I said, wait a minute. We have an abundance of leucine. All that leucine would be going to waste. So how important is leucine? Well, back to the growth again. It's the predominant, most important essential amino acid, more than anybody else. And the growth's error, and I have no criticism of any of Groth's papers, it's very clear that it needs to be interpreted, is that he normalized the leucine, and it led us astray for 70 years here. If he had normalized the leucine, it would be much more informative. So, how important is leucine? What he did, he fed newly emerged adult bees, took their weight, either their increase in dry weight or the increase in nitrogen content, at 7 days and 14 days, with deficiencies in these amino acids. And if it's really critical, that deficiency will show that they will stop growing. And which one comes through? Leucine. Boy, they'll grow for a little bit for 7 days, and then if they don't have enough leucine, man, they start losing weight. These other essential amino acids, they'll just keep growing despite not having that amino acid. So really, we need to focus on these few right here, leucine, threonine, valine, and maybe histidine right there. These are the critical ones that you do not want to run short on. You guys all still with me? Yep. Okay. I'm trying to slow it down because the translators are getting tired. And I talk fast, it wears them out. Now, let's do the same graph again. This time, instead of normalized to tryptophan, let's normalize to leucine. And look at the deficiencies. Valine, that critical amino acid, way deficient. Threonine, deficient. Histidine, deficient. Go back here. We got valine, threonine, and histidine. They're critical ones, and they're all deficient on those critical ones. Oh God, I get goosebumps right now. Because that, when I did this graph, all of a sudden I go, wow, eureka. I just figured something out right here. Everybody's been using the wrong normalization to figure out the optimal diet for honeybees. So I created that amino acid calculator so that we can compare the diets. The growth's recommendation are the gray bars, and the green are the formulated diet. So I did one for royal jelly, and again, look how closely the royal jelly matches the growth. A little bit low in isoleucine. Royal jelly is a little higher in lysine. So the growth recommendations were really deficient in lysine for nurse bee growth. And then look at a mixture of known nutritious natural pollens. Matches the growth ratios almost perfectly. So now let's look at all these artificial diets. And what I did is I ranked these from the best performing down to the poorest performing and down here with any minor deficiencies with a small circle and the major deficiencies with a large circle. You notice the pattern? The more deficiencies there were. This is all published. It's on my website. Not that you just. The more deficiencies, the poorer the growth. So I said, let's see if we can plot a correlation. Here's our correlation. So this is the ending frames of bees per treatment. And this is this, I found that the summing up the deficiencies gave me the best result. And relative to leucine, and look what you do. An R squared of 0.85, that means 85% of the variation can be explained by this variable that I came up with. And healthy bee, if it hadn't had that time mode, would have probably been right on the line, also right there. We would have got even a higher R squared. When you see it with a field trial with honeybees, an R squared of 0.85, usually you're really happy with an R squared of, 0.6 or 0.5, okay? 0.85, and then a probability of 1 chance out of 2,000. Okay, very, very low probability of this being by chance. So really shows it was that. So then I went further research and I looked at everything I could find published on the needs of the nurse bees and the larvae, and I came up with my own recommendation, higher in lysine to compare to. And that's, so you have these both on my calculator to try to figure out your own. And so I already showed you how to use this right here. I've got common ingredients here, and then you can add the analysis. So if you're living in Saudi Arabia and what you have is ground date kernels as a protein source, you can put that in there. If you're living somewhere else where all you have is peanuts, you can put that in there. So beekeeper, I made this for beekeepers anywhere on the planet to look at their available protein sources, put the analyses in here, and then they can play around and try to come up with a perfect pollen sum. Anyway, preliminary research, again, we need more files to confirm this. And I'm, again, collaborating with USDA, and I got some different phytostimulants at home, so hoping that this summer we'll get back onto pollen sub again, because I gave each of those manufacturers how to perfect for just very low cost, adding a few essential amino acids. And I don't know that any of them have done it. So one of them will probably, I already talked about their people. So I can probably collaborate with them this summer, do another trial, balancing. I want to do a diet with no pollen at all, and that's what pollen seeds, and I get it to perform well. Oh, you don't need that. Well, that was about the, okay, that's it.